All right. How you guys doing? Doing all right? Good. Getting excited for end of semester and maybe end of the degree for a lot of you. Hearing some chatter here. It's a weird time, right? It's like everything still feels normal except it's about to not. Yeah. Okay, so speaking of, I wanted to know how many of you have thought at all about the presentations yet? Anybody? Okay, I mean, I kind of expected that. I'm not by any means upset about that, but it's time, and I also need to figure this out with you because, along the way we had, well, for a little while there, we were like, hey, okay, here's the topics, pick something you can write about, and then get together in groups to talk about it. But then along the way, we said, hey, there's a project down in Nicaragua that maybe we'll do something with about this disinfection or treatment system for a family with a private well that they wanted to provide some sort of treatment. It wasn't an option to do like the full water supply. Maybe this was a family that had someone employed at Amigos or something like that, but it wasn't part of a community wider. So we never really went very far with that in terms of actually doing anything on our trip or following up. So that's still possible, but we need to sort something out because of the 4 groups now, two of the group members in group 3 have abandoned Lauren. So we can do a couple things. One, we can keep this and have this presentation as like a concept of like, hey, here's an option. We could abandon it altogether and just say, okay, we'll go back. Either of those are fine with me. And another thing we can do is we could say, well, this septic tank alternative sort of thing does tie in pretty well with the groundwater, and they could probably send one person over and kind of merge that way so that we could split it up. So I need your input though. You're saying you could do it solo. Cool, awesome. But anybody, how do you guys feel? I think it was Evan and Abby, right? Are you still interested in talking about this one? No? Going back? Okay. So we'll go back. I think that's simplest, and that'll work out well because then the timing of this schedule. So I should tell you that also. Well, I'll pull up the rubric or the syllabus in a moment. I posted a new syllabus or new version. So we will edit this timing to give you a little more space so it's not all crammed quite so tightly. We can do maybe 25. So if the presentations are about 25 minutes each, something like that, or 20 minutes, I think is the expectation I wrote. 5 minutes to transition, pull up the next set of slides, and have a couple questions. That'll get us all 3 groups done, settled, and presented all in that same day. And I think everybody should hopefully be happy with that. So, and that, my thought is that should be on the 30th. That gives us about a week and a half. So let me pull up the syllabus and I can ask you about that. Okay, so this is kind of the outlook for the rest of the semester. Kind of filled in where we've been. I also posted update to this to Moodle, all the lecture slides we've done so far to Moodle. I also realized there was a lot of the writing tips, a lot of links about all the stuff I critiqued you on. I had not activated them, so those are all now visible on the Moodle page. So under the writing assignment topic, check there and you should have a lot of good resources. Also the slides that I covered that day and in the lecture itself, please go back to check that out because it, I think would be very useful. If you have any questions like, why am I getting comments on this thing? Or what do you mean when you say check the caused by shortcut for because of or due to, right? If I wrote anything like that and you're not sure, good resources there to take a look at. Okay, so today we're going to talk about LED systems a bit. Hopefully I'll be able to wrap that up a few minutes early and let you break out into groups, start planning for your presentation. Next time I've got some slides for innovative toilets, and then I thought that it might be nice to kind of circle back a little bit to looking at development now that we've covered a lot in the class, water treatment specifically. How would that all fit into our wider view of development kind of globally? Group presentation then, and then cover some practice problems, the type of things that would be on the exam, kind of do a bit in class, kind of look at a few problems, discuss them, kind of see where everybody's feeling about that. But if you'd rather, I can swap these two. That's fine with me. Your paper should be due that Friday the 3rd, so keep that in mind as well. Any thoughts, questions, feedback on all that? Okay, feeling all right? Yeah. Yeah. Oh, okay. Okay. Is that during this time period? It's what? Okay. Do you feel like you need to prepare a lot for that? You're giving the same posters you're giving tomorrow, right? Okay, so you should be prepared by tomorrow for this one, right? Okay, but let me know if you feel like this is too much. But I think you shouldn't need too much preparation for that. Okay. Yeah, we can talk more though, and I'll let you guys know if we need to move maybe one presentation to this date or something. But I think Evan can handle it. It's just my thought. He climbed the volcano after all. He did it. Okay. All right. So if you need to talk to me about any of that, let me know. Okay. So today, all that aside, I'll update this in Moodle as well. Now that I changed this a bit. But everything else I think should be pretty much current in Moodle. So today, we're going to talk about another research topic that we've been working on with a variety of students for several years now. And we have a little bit newer data now. I haven't incorporated all of it yet, but we'll kind of talk about where it's going a bit. And ultimately, I hope that this can be a nice tie-in to say, hey, here's the type of technologies that are developing, at least in my side of the water treatment world, that may be able to be applied for kind of developing contexts. That's sort of my aim here. So a lot of this is stemming from a project that I received some funding for that's essentially aimed at accelerating sustainable water treatment using LEDs. And I'm going to say smart LED treatment, because we can do some clever things with LEDs that we can't do with regular lights. In particular, we're able to turn them on and off instantly. That doesn't sound like a big deal, but practically speaking, if you only need the disinfection for some times of the day, that's actually pretty helpful. And then ways to control it. We'll get into several aspects of that, but they're just a lot easier to control and use, and they give us more options in terms of the chemistry behind it, or the photochemistry, than we had with just our kind of conventional lamps, light sources. Okay, so what we'll talk about today is, well, why do we care about these LEDs? So what? We have new lights. They shine light, big deal, right? Why might that be important? What about disinfection? How does that affect UV disinfection? Can it be a beneficial thing to change the light we're using? Could it be maybe a bad thing? Can we prevent bacteria from being able to repair themselves? That's been an ongoing kind of target for research. And then on the pharmaceutical side of things, can we kill these pesticides? Can we destroy the Tylenol, the caffeine, the heroin that is discharged into our wastewater? before it gets into our environment, or if we have some contamination coming into our drinking water supply, can we destroy that with this kind of process? And what are these new lights? What do they do for us? Okay, so conventionally, our typical UV lights were mercury-based. Still many are, most are. This would be some sort of mercury vapor at some pressure, so we have some amount of mercury vapor inside this quartz tube, and you send an electrical arc across it. What ends up happening is you get the mercury kind of excited, and then it's going to emit a characteristic signal of photons based on the energy level, that energy transition between the high energy state and the ground state. And that's just going to keep happening over and over again, but these take a little bit of time to warm up before the mercury vapor is the right temperature and the efficiency is happening correctly. They also require a ballast to manage that stuff. That also needs a little warm-up time. And it needs this fragile quartz casing so that the UV light can go through it. Turns out UV light doesn't go through glass very well. The silica oxide bond in glass, just like our window here, that's going to absorb all the UV light, at least in the germicidal range. Probably all of the UVA light too. So if you ever thought about it, you've never been sunburned by going on a road trip in the car with the windows up. The only way you're going to get a sunburn in the car is if you have the window down and the sun's shining right on your arm, let's say, or whatever. So you've never even probably thought of it, but why aren't you getting sunburned? Well, that glass is absorbing that UV light. So we need a fancy quartz casing for this, and probably some extra protection around it. We usually use a quartz sleeve so that we're not breaking the lamp if something breaks. It's that sleeve, and we're not getting junk on the lamp itself as water passes through and organic matter starts sticking to it and these photoreactions happen. You end up getting some gunk accumulation on your lens, essentially. So instead of applying a UV lamp just by itself, we actually use, and these are fragile enough on their own, we use some sort of a casing. Also needs to be quartz, so it's also fragile and expensive. And then you have to clean it, which is difficult to clean because you really have to scrape at it, so they often break when people, the operators are cleaning them. So it's really not ideal. These quartz sleeves are actually a major expense for UV operation in practice because they break so often. And the maintenance takes hours and hours of labor. It's labor intensive and costly because a lot of times that labor ends with a broken sleeve. So on the other hand, if we use these LEDs, and we've got a photo from our lab here, we can potentially set up an array, and each one of these are like a little point source that's illuminating, so it's a bit of a different apparatus. Maybe that could be an advantage in some ways. They're also going to use a different form of electricity, so DC direct current power instead of alternating current that the mercury lamps use, the fluorescent type. That can be a little bit handy in certain circumstances if you're going to use a battery. It's a little more efficient because that's what they provide is direct current. If you're going to use solar power, that's also direct current, or typically maybe charge a battery, which then goes here. So you get some potential energy savings there, especially for portable kind of resource scarce situations. In addition, if we shift the illumination color or wavelength a little bit, we can start using something else other than quartz. Maybe it doesn't have perfect transmission, but some types of glass or even some plastics can allow light through at slightly higher wavelengths. So we'll talk a little more about that in a minute. The other thing is we can make them blink and flash and do all these fun things, right? So if you've never seen these Christmas light shows, I think you could do this with like the incandescents as well, but this just kind of makes me think of like those light shows that are crazy and usually tuned to some music, right? So we can blink them on and off. And aside from looking cool, perhaps there's something else we can do with them that's actually useful on a technical sense. So we have a few hypotheses we've been working to test, and we've seen some data so far that looks promising. I'm going to show you some of that. We're not positive that this is the case, but we are thinking that maybe if we are pulsing our lights, we can get some special outcome, because as soon as light's exposed, or let's say some photoactive material like we talked about last time, or something that we are causing to split up, like hydrogen peroxide, to create radicals, as soon as the light's shining, action is happening. So if we shine the light and then stop it, if there's special things happening right at the start of the illumination, then we can repeatedly start that and not waste so many photons. So there's some theory that maybe if we're doing on and off, we get kind of a spiking sort of a deal, and maybe our overall average reactive stuff might be a little higher than if we just kept it on the whole time. Part of that's because we might waste some photons. And in addition, let's say we do create some radical, maybe continuous light could shorten its lifetime by causing it to react quicker than we want it. So there's a few thoughts there that maybe we can do something special. Another idea is that we can actually select different colors or wavelengths much better. The LEDs give us many more options. So if this is a mercury lamp, characteristic low pressure mercury lamp, and then a medium pressure is kind of like this little line I drew last time. Well, the LEDs, we can select several different peaks, even some out in the UVB and UVA territory, of course. And so we have several options. We can center them at different places. This may help us with a wavelength that, let's say, targets, instead of hydrogen peroxide, maybe we're trying to split ozone or some molecule called peracetic acid. Different things will have different absorption spectra, so maybe we can target something specific and pair our light source to that molecule that we're making use of. So that could be a pretty good advantage, because typically we were stuck using this, which is pretty good for the germs, but you need quartz. So if we can just shift it over to, let's say, this blue one, maybe we don't need to use the quartz anymore, because that type of light at the 280 nanometer range can pass through certain plastics or even a silicone covering. Okay, so a little more on that. A few other things that why just simply adjusting that wavelength might help a little bit. This is a bit of a caricature. The mercury lamp and the UV lamp, these are actual data collected from our lamps in the lab. But then I sketched on top of it kind of a typical germicidal curve. This is essentially where DNA absorbs. We talked about this, I think, a little bit last time. So DNA will absorb light approximately in this sort of a range at that amount. So we see the mercury lamp does really well. It's emitting strongly right where it's absorbing pretty strongly. However, this UV light, maybe there's some advantage, maybe it's not quite as good, depending on how good I drew that. Probably, I think that this curve actually looks a little more like this. So maybe I exaggerated the effect here a little bit, but it's close. If we shift this LED over to a 265, it might be actually ideal, which those do exist. So some of these might give a nice benefit just for the curve, but in addition to that, we also typically have other things absorbing light in our water, causing attenuation that we don't want. So we've talked about this before, but if we take a light and shine it through some colored water, it's absorbing some of the color in the light. That's why we end up seeing, like, if it's my bottle here is absorbing light, so that all we're seeing is blue coming out of it, right? So whatever colors needed to be removed to get blue there, those are being taken by the plastic. But the same would happen if you had tea, right, or Kool-Aid or something. That color is being absorbed across the distance. And one thing that happens is dissolved organic matter. Maybe this is natural organics that are just in the water. Maybe we're treating wastewater, so we'd call it bacterial organic stuff. Whatever it is, it tends to absorb light, and it tends to absorb pretty strongly the lower wavelength we go. And so just shifting it from here over, and if we just shift over to the kind of the wavelength here, we notice we have a nice drop in the amount of interference from that organic matter. So there's some potential advantage just simply from that. So several reasons here why we may get some benefits from doing this smart disinfection. Okay, so I want to talk a little bit about how exactly the disinfection works so that we can talk a little bit about, well, what happens if changing the wavelength here from mercury over to some LED, is that going to cause a difference in the actual photochemistry of what's happening to kill the microbe? And it turns out that there's probably some differences, and there's still a little bit of a question of how important is that? Can it be an advantage or a disadvantage? So that's what we're talking about next. So the inactivation mechanism for direct UV disinfection, this is that UVC stuff that the atmosphere blocks for us. So if we were up in the space station, we'd worry about it because it's going to, that would be quite the nasty sunburn that included a lot of DNA damage. Not the same kind of sunburn we have down here. So what happens is there are these base pairs with the DNA. Normally they're, I've used the zipper analogy before. It kind of acts like a zipper where each prong has some sort of base pair and it's going to match the next one and be able to zip and unzip to replicate the DNA or make copies or code certain proteins to be made, all that good microbiology stuff that you've learned about. Well, when a UV photon hits the DNA molecule, in particular areas where you have two thymine dimers next to each other, that will cause a cross-linking. So instead of just having these two base pairs next to one another and something else on the other side, now this cross-link bridges them and sticks them together as now, instead of kind of acting like two different molecules that are just stuck on this backbone, now it's like there's just one with their that are one molecule that's linked. So that causes a big problem for the DNA, for replication, for any of that. Now there are cellular mechanisms that can go back and repair that. So it's not the case that as soon as we get any cross-linking, it's just a completely broken DNA. But it needs to be repaired. So if you're delivering a lot of that, and perhaps a cell could live with one DNA molecule broken, right? Maybe it won't function as well, maybe it'll die sooner or something, but chances are good just one spot is probably not enough to prevent replication and growth. You get a lot of them though, and if you get too many to repair, then there's no chance for that cell to continue on or to replicate. It's interesting here too, because it's not necessary for the organism to die to be disinfected. If you remember, we talked about all we have to do is prevent it from being able to replicate. So that's the target here. Okay, well, turns out that a lot of microorganisms, and I think larger organisms as well, have developed some sort of repair mechanisms for their DNA. And one in particular is called photolase or photolase. That's this enzyme here, and it's actually activated by blue photons. So if a microorganism is hanging out in Lake Pontchartrain or something, gets exposed to some UVA, UVB, maybe some DNA damage happens. It's not super efficient. If we go back here, this germicidal curve, it's kind of tailing off in the range where we might get a little bit of sunshine down in this range. Remember, this green curve that's just my arbitrary estimate with a PowerPoint sketch. So sometimes they'll have a little bit of damage from sunlight, is the point. And probably occasionally something can happen with UVB I guess that is UVB. So we'll get a few damages. And so they kind of have a built-in repair system so that when they're exposed to that kind of light, they're also going to be typically exposed to blue light and UVA range light. So less intense photons, And this molecule is actually activated by that light to go and fix these dimerizations. So it's a really neat molecule, and this the exploration of it actually resulted in a Nobel Prize from these researchers. And it was very interesting because they first observed it in 1978, and it was kind of an accident. They were just doing some disinfection experiments, and then they noticed, wait, why did these samples die off this much, but these samples died off less? And then they realized they had left some of them out in the laboratory light while they were incubating, and some of them had been in a dark cabinet. And so then they're like, hey, something's happening here that's repairing these according to exposure to the laboratory light. And so that got them researching. They eventually realized, okay, there's some sort of enzyme here. And then really what took this research further was they wanted to get a therapeutic drug from this. You know, if we think about penicillin, we get microbes to produce a bunch of this I don't know if penicillin's an enzyme, but a bunch of this biomolecule, and then we can harvest that, and we have a great antibiotic. It's kind of natural source sort of situation. And so the researchers was like, hey, maybe if we get a bunch of E. coli or some other organism to produce a whole lot of this photolase enzyme, then we could harvest it and use that to treat a sunburn, right? To repair all these cells that have been damaged, something like that. So it was a great idea. And then as they were researching, they found that they were never able to do it. Turns out there's only like 3 to 5 of these molecules per cell. So there's just very few of these molecules in a given bacteria cell. Humans do not have this enzyme. We have some other repair mechanisms, but not this one. So it turns out only 3 to 5 enzyme molecules per cell. So it's a little unfortunate on that front, but they did ultimately learn quite a bit, characterize this mechanism. And so you can see the reaction here kind of pictured nicely. So we have 2 thymine groups here. This is what they look like when they're healthy and normally sitting on the DNA, the DNA backbone. That's this here. Once you hit them with the UV light, you'll get that cross-linking here, shown in red. But then the photolase enzyme plus blue light can revert it back. So it's essentially just like this little, I don't know, one of those train carts that's going along the train. It's powered by the blue light to go and repair. And if you don't have that light, it's not moving along the train tracks. You might think of it that way. So these are just running around in the molecule doing repair the whole time. So this kind of begs the question, thinking about now that we're going to use a different type of UV light for disinfection, does it matter? Are we doing other types of damage? There are some other dimerization reactions that can occur. Are we going to damage the photolase enzyme itself or some other aspect of the cell when we're doing it? So we recently had a publication on just that, and so I'm going to show you some of that data here. So the real question we started with was, if we have 254, that's the mercury lamp, versus an LED, are those damaging the cell differently? Or is there some other mechanism that might be important here? And if you look at the wavelength we're dosing with, there's a decent chance that we are doing at least some extra damage over here. It's not so good with the germicidal stuff, with the direct dimerization reactions, but it's probably promoting more reactive oxygen species production and oxidative stress type damage. So the kind of stuff we typically receive with sunburns. So there is pretty good reason to think that could happen. So just a little calculation or bit of data up front here. If we just check the disinfection by itself, we see something pretty typical. The mercury lamp on a photon basis is a little better, but it actually, this is a little misleading, because this is, if we're checking the photons, we actually should only count the photons that are useful for that UV damage. And so we should actually reduce the amount of photons we counted here by something like this. So somewhere around there would be the cutoff. Oops. Can't draw. So if we calibrated like that, we're just simply counting the number of photons that are causing damage on the x-axis, because it's kind of like how intense the light is times the time that it's shining. So if we calibrated everything perfectly, we'd see these are about the same. The wavelength doesn't matter too much. So, but if we count all of the light emitted, we see the mercury lamp is a little better because all of the light that's shining is damaging the DNA, whereas not quite all the light for the UV 278 was doing the damage. You see another typical thing up front here. We have a little area where it's not happening very quickly, or it's not immediately going into a first order destruction or killing of the microbes. They have some inherent resistance. It takes we have to cross some threshold before there's enough damage for the act to actually kill them, right? So a few dimerizations would probably be okay for them. They could repair. Just like, if we go out into the hottest, brightest, sunniest day we ever experience here in Louisiana, it's okay if you go out there 5 minutes with no sunscreen. That's probably not going to do enough damage, even if you're the most pale person in the world, right? You're probably not getting a sunburn with 5 minutes. So that's a dose response here. But otherwise, that was pretty typical. So we went on and we said, okay, we know there's some repair mechanisms. We want to know, is it different? So the first thing we checked was, is it different in the dark condition? Our kind of hypotheses are about the photo repair condition, but we were also curious, well, what's the baseline? Is that any different? And actually we found that maybe there's some difference. If we check, we gave the bacteria 2 different conditions to try to repair in the dark. So we'd first shine the light, and then we'd close it all off and make it dark. And we actually left them in solution, and in one case, the highlighted case here, this would be the green triangles and the purple upside down triangles. Those 2 cases were the mercury versus the LED with culture broth. That's the CB. So with culture broth, seemed like that gave the bacteria a big difference here in what happened, because with the UV 254, the mercury lamp, they really didn't grow back too much. They didn't recover very much in the dark. Whereas the UV 278 ones, they were actually able to recover quite a bit in the culture broth system. All the other cases where there was no culture broth, there was maybe some organics, they all seemed pretty similar. So something interesting here, seems like if they had access to good nutrients, they could repair pretty well from the one versus the other. So kind of interesting there. We don't have an exact reason, but maybe that's a function of some of the damage. The oxidative stress was repairable from this dark mechanism with access to food. So something like that. So we went on to test more so on the photoreactivation, so when they're exposed to light afterwards, given different amounts of repair light and different types of repair light. Now where this becomes relevant in the real world is if we were to treat some water and then put it out, if I were to use UV disinfection for my water bottle, and then I treated some water, I drank a little bit, and then I set it out, and it's sitting here under the light or under the sun for a couple hours, are they all going to repair themselves, or are we safe? There's a question there. Or if we're disinfecting some wastewater and discharging it out into a river, and then it's going to go, and there's some public use areas, and we monitor, we try to keep our waterways safe for contact recreation, right? That's a big goal of our wastewater treatment. So we typically want to reduce that pathogenic organisms or the indicators for them. Well, once they're out in the sunlight, what's going to happen, right? Are they going to repair themselves? To what extent? How much does it matter if they have UV light with the sunlight or not? So we went out, set out to test, okay, how about high intensity light photo repair light. And what we're using here is, it's like one of the, just on the verge of a black light. You know, it's a 395 is kind of where those are centered. 400 is kind of our cutoff for visible light. I should have brought, I have a 395 LED flashlight. I should have brought that in. But you can see it looks violet. Of course, you probably can't see the photons down here that's emitting, but it's definitely like you've probably seen black lights before where you can kind of see them purplish and it sort of makes your eyes a little sore, understandably so. But it will also cause your clothes to fluoresce or different things, and you know, it's cosmic bowling or whatever. So that's the type of light we're checking, and we just basically did 3 different intensities. And then in addition to that, we checked 3 different types of light. So the kind of the 365, that's your conventional black light. This 395, which is closer to visible, probably that laser, like the violet laser pens, if you've ever seen those. And then a generic kind of bright white light LED mixture. So that means, you know, it's kind of like the indoor lighting replacements, that type of LED where it's actually got lots of colors in it, but it comes out pretty much white. So those are the 3 different conditions of the light types. So we did a light type and we did a light intensity comparison for how are things repairing. So lots of scrambled looking data here, but essentially we were looking at over time, how does that intensity affect it? And it turns out, well, if we're using this UV light, if we give it a lot of intense light, they can repair quicker. So if we have a lot of high intensity, over 3 hours, they actually repair quite a bit, but then they're getting stressed more than in other conditions. So it's kind of like if they're getting high intensity, and you kind of maybe expect this, they're also then dropping faster than the others. If we put it on a photon basis, you can kind of see it a little better. So if they're getting really high intensity photons, they repair up pretty fast and then die off pretty fast. That held fairly true for both when exposed to the LEDs or the mercury lamp, and that's what we're exploring is there some difference to how these are impacting it. But in general, it seemed, at least in this case, the UV LEDs caused the bacteria to be a little more susceptible. So that might actually be a good thing in terms of if under natural conditions we disinfect with a UV LED, then given, if we give them then sunlight, for example, they might repair kind of quick, but they're also going to get stressed out really quick because they've already been exposed to some stressors like that. So maybe we could actually add some of that UVA to our disinfection in the first place and maybe get some advantage there. Okay, so then we looked at the light source instead of intensity, and kind of as we expect, the UVA, that 365, that's harsh, that in both cases, they were dying off faster that way when exposed to that more harsh light. One case we saw with the white light, it's kind of an interesting effect here. It just kept repairing. There was never a spot. I mean, this is still a like, I guess by the time we get up close to the 0, we're near where we started, because we enacted kind of a 3 to 4 log inactivation in the first place. So that one actually came back almost completely repaired, and that was from the 278 under just the visible. The mercury lamp was pretty close also. So one thing we see though is if there is that UV exposure for the photo repair, it's definitely stressing them out after helping them repair. So it's kind of like it's repairing it, but it's also causing damage over time. So kind of interesting. Tells us maybe how to manage post-disinfection conditions. In all, one of our overall hopes here is to be able to model using this fancy equation some researchers developed. Basically a mass balance on how many bacteria are viable after exposure given some light. And if we put all the parameters in, we should be able to model the survival fraction at some time afterwards, given some intensity of light, given some intensity of the initial disinfection light. So there's all sorts of parameters here. It gets a little challenging, but we were able to use that model and come up with some parameters to try to match them with a previous study. So that was something that we pushed forward and continued from there and checked a bunch of different reactivation light intensities to see if we could model those and get parameters for each of them. So we're still working to wrap this up and get this part published, but a pretty neat concept that maybe we can use this model then to predict what's the damage going to be in the environment. Meaning we use UV to disinfect, now the bacteria are released from our effluent. Given some water body conditions and solar conditions, how much do we expect for these to repair? So we might have designed our system to achieve a 3 log reduction, but effectively we're going to get a 2 log reduction given the conditions. This is the type of model that would help us plug that in and check that risk factor, check that regrowth potential. And maybe all we need to do is blast them with some UVA in addition to the UVC. That might also be a solution to kind of overcome that, exhaust their repair, and break them in some other ways. Okay, so that's kind of the disinfection portion. Some neat topics there, potential for exploration of mitigating that regrowth. Didn't really go too far into it, but I think we talked about it a little bit. Maybe this in the disinfection front, there's also opportunities for small devices that are much easier to work with. I think I have a picture later where we had a 3D printed lid and just stuck a LED under it, connected it to a battery, and disinfected my bottle, right? I think there are companies making things like that already. So it wasn't like an invention per se, but it's something like you can kind of put together something like that rather simply. Okay, so next up would be advanced oxidation processes. What are we doing with that? What are the new opportunities? How does that look? So chlorine in particular is what we've looked at so far. And the reason for this is because typically chlorine, we kind of know, it's obviously an oxidant, just like peroxide or ozone, but it hasn't been typically used as an advanced oxidation process. So hydrogen peroxide plus UV, ozone plus UV, these are common advanced oxidation processes. In fact, you can even do hydrogen peroxide activated by iron. That's called Fenton reactions. All of these are pretty common advanced oxidation systems. But chlorine never really got much use, in part because 254 is the wavelength for the mercury lamps that we typically use. Well, sorry, I just wrote all over that spot. I thought I might, yeah, there we go. Okay, so I'm gonna erase this. So the typical UV spot where we would normally activate chlorine, the molecules don't absorb. HOCl and OCl minus, that's kind of their weakest absorption throughout the UV range. I'll just write a note. Peroxide and ozone are kind of the more common ones. Chlorine really hasn't been used a lot. Now, however, since we have more control over what wavelength we're going to use, we can select a UVB LED, so 305 nanometers, or that 278, and target the chlorine compounds to cause photolysis much more efficiently. There's some caveats in terms of quantum yield and things like that, but it actually gives us a lot of control because then we can target and zap one of these based on the pH maybe, and get specific outcomes based on their degradation. And those can then go and destroy the pharmaceuticals, the pesticides, things like that, in addition to doing a great job killing microbes. So it's kind of an advancement onto the disinfection. So when I say we can do it with a lot of different reactions here, got a lot of different radicals that are in play that we've identified. Not meaning for you to pay too much attention here, but one thing you'll see is we have HOCl plus photons gives us chlorine radicals and hydroxyl radicals, or we can take OCl minus and photolyze that and we'll get different radicals. And those different radicals from each of those end up going through lots of different other reactions based on where we're at. So we do end up with very different systems based on the difference in wavelength or the difference in pH that we're using. So it's actually pretty neat because then we can design our system in a particular manner. Now, something that we're eventually going to explore is how about when we have chloramines, which is pretty much any time we're doing chlorination with either wastewater that's going to have some ammonia, some nitrogen in it, or maybe if there's a bit in your drinking water, or maybe you add it on purpose if there's some, we typically do use a little bit of chloramine as a residual for our drinking water. Okay, at any rate, we have these cool new options for the LEDs in addition to the old school mercury, and all of that gives us some neat options. So we get wavelength dependent species that we can use, and maybe that helps us overcome some of the typical challenges for advanced oxidation processes. Well, one of the challenges is in typical systems, you probably have some organic matter. Hydroxyl radicals like to react with just about anything, so a lot of them are wasted to just reacting with organic matter that kind of didn't matter, right? It didn't have anything to do with the safety or the whatever. So we just end up using a lot, whereas if we select, let's say, this system can actually produce ozone specifically, if we cater it right. That might be a good advantage. I mean, you might think to yourself, well, why not just use ozone itself? But there's actually some benefit here because chlorine is a bit safer and easier to work with. So perhaps we can get just the right amount of the radicals and target accordingly. Or maybe the pH of the water changes or something, we adapt. The fact that we could switch from one LED to the other instantly based on a change in the incoming pH, for example, could make this truly a smart system where we have a sensor. It says, hey, the pH just bumped up because something weird happened upstream. We're going to adjust the chemistry just like that and account for it. So some pretty neat options there. Okay, so I mentioned pH, wavelength, and ozone just now. There's actually a bit of a scheme. This is a good paper several years ago by this group down here. And we see that if we have higher pH or higher wavelength, we tend to get more ozone. Whereas if we want the stronger, less selective radicals, if we want hydroxyl radicals and chlorine radicals, we get more of those if we go in the other direction, right? And so there's some option here, either to account for the pH that might be changing, or to simply say, can we alternate back and forth with the wavelengths to get some of both during our reaction. So there's some really neat potential opportunities here. Maybe we give it 20% of the light is getting the strong stuff, and 80% is getting the more selective stuff. Maybe some ratio like that would be ideal for a particular system. Okay, so that's kind of the motivation for a whole bunch of data right here that I'm showing. Not meaning for you to look at it too closely, but what we're doing here is we're making these plots where we're checking the kinetics, right? We've seen this quite a bit, hopefully, where we have our dc/dt. We'll have some rate constant, something like this, to the first power. And so if we plot time versus the natural log of c over c naught, we'll get a straight line there, and that will be k. This slope will be k, right, for this reaction. So this is first order kinetics put into place, and we're checking the destruction of some pharmaceutical in different conditions. So when I show you all these plots, you'll notice the y-axis is just that, natural log of c over c naught of some sort versus time, and we're kind of exploring how quickly is this happening. So that's kind of what's going on. And then in some of these plots, we will actually take those those K values and just compare the K values to say, okay, this one's faster, better. So that's kind of where most of this analysis is coming from. So one of the first questions is, can we circumvent the challenge of organic matter? So we have a few different organic matters that we've been playing with. I talked about this a little last time with the colloidal particles, the hydrophobic, transphilic, so they're like partly hydrophobic, and humic acid. So we're checking different pHs. So we go from, for each of these, we go from 3 to 5, to 8, and then 10. And we're just taking a look at how much of an impact. So if we have no organic matter, we have that at that 0 line, that's our typical destruction of acetaminophen. So Tylenol. Destroying that, also known as paracetamol. So we're destroying that, and at default, it's up at that 0 line. And then we see, okay, if we're operating at pH 3, that tends to drop our efficiency by 50% or so. And we see some dependence on the type of organic matter there. It turns out that pH 3, that's going to end up being mostly hydroxyl radicals and chlorine radicals. Whereas, so probably 3 and 5. Whereas 8 and 10 are a little bit more ozone and some similar compounds or reactives. So what we notice is, well, some of these we're actually doing really well by selecting these higher pHs. Some of these, well, really all of them, the system seems to be reacting better that way. So it seems like, at least for acetaminophen, might be different for a different compound, but at least for this case, ozone is doing really nice work for us. So in this case, we'd want the high pH and or higher wavelength if we were selecting based on that. Okay, so we also confirmed that, yeah, we really are getting ozone here, in part by just taking out the oxygen from a couple of these. And at pH 8 with oxygen, we get a nice destruction, whereas at pH 8 with no oxygen, nothing happens. So an oxygen was a key component to that reaction. So it's pretty neat to confirm that, and also to see that at pH 5 with oxygen, we're not getting a whole lot of destruction. So really just kind of confirming that, yes, this is ozone that was doing the action here. Okay, so I already talked about this, that option of going back and forth between selective and non-selective. So this is the kind of the scheme of the next set of data, is we actually went ahead and tried that to say, hey, does some combination of these, is that better? Can we do some sort of enhancement here? This also kind of gets to this pulsing again, where maybe something special is happening if we're giving it a brief amount of time with one than the other. The idea being, if we're constantly on, maybe this is kind of typical amount of radicals here, whereas maybe we can elevate it if we're doing this kind of cyclical thing. Okay, so here's, again, destruction of this acetaminophen. And on the y-axis, we've got the observed rate constant. So again, that's the, on our plot, that's the slope. So we observe some rate constant, and we get that value, and then we're placing that value on this plot. In this case, what we're looking at is just pulsing the light on and off. And the DC here, that stands for duty cycle. The duty cycle is simply 0.5 is 50%, so it's on half the time. You have it on for a second, off for a second. On for a second, off for a second. And then the frequency here, 0.1 every, so 0.1 means we have 10 times a second. So 0.1 per second. So this is, should be 10 hertz, then 100 hertz, and 1,000 hertz. So Hz is the abbreviation for hertz, and that means cycles per second, right? So it's, this is what it would look like. Okay, so in A here, we have this chlorine and the UV, and A is giving us the pH 3 values, and B is giving us the pH. Oh, excuse me, we're doing different wavelengths, not pH. This is all at pH 3, as is already written. So this would be the UV 278, and this is the UV 305. So the 305 should give us maybe a little bit more of the ozone, but at pH 3, it's mostly going to be the hydroxyl regardless. Okay, so all that said, what I really want to highlight here is actually just this gray line across, or the black line across. That's what it looks like if we have the lights on the whole time. 100% of the time, they're just on. That's the line we get. Now, in both graphs. So it appears that if we have pulsing, maybe around 40 to 50%, the green and the red here, that's actually optimal. So it seems like our rate constant, and this is not even accounting for the energy used, this is just the raw, how quickly it happens, is actually seems to be improved in both cases if we don't use it on the whole time. So that's pretty neat because it's a little bit of evidence that, hey, maybe the pulsing system is actually, maybe there's something to that, and we could be getting some benefit there. So we've got a little more to do to confirm that and to see really, okay, how much error are we expecting here, but it is pretty encouraging and consistent seeming so far. Now, if we do that at pH 8, this is a bit of a different scenario because we're probably mostly getting ozone instead of mostly getting those other ones. And here we actually see even more improvement compared to the baselines. The thinking here is ozone lasts a little bit longer and might absorb our light. So if we create a bunch of it and then pause, maybe that's actually really beneficial because then it has time to go and react with something before it gets blasted and split apart again. Okay, so something like that could be happening, could be pretty neat. We went a step further and we said, well, what happens if we use this UV 278 and the UV 305, try that alternating. And so here we have a graph showing that with X, meaning some portion of it is the 278 versus the 305. And essentially the green, X equals 1, we have 50% of 278 and then 50% 305. The lines across are just what would happen if we had just one or just the other. And in both cases, we see, you know, what we're looking at is the combination of the two is better every time. And it turns out that when our fraction is 4, of all the ones we tested, that seemed to be the best. So that means that's 4 times the amount of the 278 compared to the 305. So found some ratio, seemed to be quite beneficial. Sum of both is the answer. And we checked that across several different frequencies and got basically the same result. Okay, so pretty interesting there. It does seem like combining it is giving us some special effect. We've got more to study to say, well, what is actually happening here in terms of which molecules are at play. Okay, we did that at pH 3 and 8, and actually at 8, we did not see quite as much of a benefit. So that was the thing. At pH 3, it was a good benefit. pH 8 seems like just purely the 305 light would have been better for the most part. Okay. So future goals include doing this with chloramines. I already thought that the chlorine chemistry was pretty complicated. As soon as we're going to chloramines, it's even just a whole lot more so. So that's something that I hope I have ambitions to study, and it would be very relevant to a lot of systems because we often have ammonia with our treatment, or could easily have it if there's some benefit, and a lot of interesting things that might come of that. Are we going to create some interesting byproducts? Are they going to be toxic? We're going to release some nitrous oxide, which is a greenhouse gas? You know, what's going on here? What can we do about that? But that's future. We're not there yet, and might not be for some time. Okay, so just a couple quick pointers of what this might look like. Point of use devices is really our target. And some of you might recognize the settings from this photo. This was a year ago when I visited Nicaragua under the pavilion there. And I brought this little system here. This is a Del Agua purification system. It's kind of designed to be at like a household tap to just add a bit of UV disinfection in line with your system, just for a little extra safeguard. And essentially, I was just pouring some water through and seeing how well it disinfected it, collecting it in a bottle. But it has a little flow meter in here and a little UV chamber right here. And so as the water is flowing through, it'll kind of detect that the water is flowing, and then it will activate the light here and let water go through. There's a little indicator LED here to say, yes, it's on. And so we noticed that if we got the flow running properly, then it activated and it was purifying the water. So we collected some field data there last year, kind of as a somewhat practice, also just kind of seeing like, hey, is this going to work? You know, how well is this working? We took some nasty water. Actually, some of the water we found at first wasn't nasty enough, so we had to find something worse. So ultimately, I think we collected some water that was in like a feeding trough kind of situation where animals were using it in the middle of the hot day, all sorts of leaves and things in it. It's clearly dirty water, so we found something that could do it. And then I mentioned this was this water bottle here. I did the ultimate no-no and took it into the lab and filled it with some E. coli. Don't tell EHS. You know, they might not like that. Of course, I thoroughly bleached it afterwards and like scrubbed it clean and dishwashed it and probably did that again, and totally safe after all that. So don't do this at home, but I did it. I did a no-no. But if you look here, here's that battery. We've got the wiring and stuff just clipped on, and right here you can kind of see the LED shining. The plastic's going to block all the UV light, but we put a bunch of E. coli in there and watched, and we were able to get, I think, a 2 log reduction, so 99% in like 5 minutes or so, and maybe went a little further even. So it was kind of fun. It was a high school project to put that together and assemble that. Okay, so I'd like to thank a lot of different students that have been involved with this kind of stuff. And over the years, and more to come. Yeah, any questions on this? All right, so why don't you take 5-10 minutes? You've got about 10 minutes remaining. Break up to your groups and make some plans for that to teach us some lessons in a couple weeks. Do you know your groups? Okay, let me pull that up.